Hello, meet my cat, Oliver, a Scottish fold. At first glance, he's like any other curious cat, playful and energetic. But if you look closely, you'll notice something that sets him apart his ears. They fold forward, unlike the pointed ears of most cats. What makes his ears unique is not just a physical difference, but a fascinating story. I've always wondered why, so come along. Let's unravel the mystery of how his ears came to be and explore the science behind it. The story of the Scottish Fold begins in 1961 when a white barn cat named Susie, with unusual folded ears, was discovered in Scotland. This mutation fascinated the first breeder, William Ross, who began to breed Susie's kittens, creating the Scottish Fold breed we know today. However, I'm not entirely sure how Susie's ears came to be. Let me ask my mom, who works with animals. Hey mom, can you explain to me why Scottish fold cats' ears are like that? Of course I can. In a typical cat, the ears stand tall and firm, supported by strong, flexible cartilage. This allows them to rotate and capture sounds from all directions. But for Oliver and many other Scottish foals, this cartilage is different, much weaker, causing the ears to fold forward. This change is caused by mutation in the TRPV4 gene, which codes for a protein that is crucial for cartilage development. But how does this happen? To answer that, we need to look at the genetics inside all of our cells. Every organism, from humans to cats, has DNA, our blueprint for life. This DNA is composed of sequences of four bases: adenine, thymine, cytosine, and guanine. These bases pair up in specific ways to form genes, which carry instructions to make proteins. Proteins are the building blocks that shape our traits, like eye color, hair texture, or ear shape. But how does DNA become a protein? That's where transcription and translation come in. Transcription is the process by which a segment of DNA is copied into RNA to express a gene. It takes place in the nucleus of eukaryotic cells. This is because the DNA must be transcribed into messenger or mRNA before being transported to the cytoplasm for translation into proteins. The enzyme RNA polymerase unwinds the DNA helix and synthesizes an RNA strand, using the DNA as a template. RNA serves as a temporary copy of the genetic information, allowing it to leave the nucleus and deliver the message to the ribosomes, where proteins are produced. This enables the cell to use the information in DNA without directly exposing the original DNA. Complementary base pairing refers to the specific matching of nitrogenous bases between DNA and RNA. In DNA, adenine pairs with thymine and cytosine pairs with guanine. In RNA, adenine pairs with uracil and cytosine still pairs with guanine. This ensures that the genetic code is copied accurately during transcription. An easy way to remember this is that transcription is like copying down a recipe from a cookbook onto a notepad so you can take it to the kitchen and use it to cook. The cookbook, DNA, stays safe in the library, which is the nucleus, while the copied recipe, mRNA, carries the instructions to the kitchen, ribosome, to make the meal, which is protein. Translation is the process where the cell uses mRNA to create a protein. It happens in the ribosome, which acts like a factory where proteins are built. The process involves three types of RNA: messenger RNA, transfer RNA, and ribosomal RNA. Each has a specific job to help create the protein. It carries the genetic instructions from DNA to the ribosome, where it provides the blueprint for making a protein, while tRNA brings the correct amino acids to the ribosome, matching them with the mRNA code. rRNA is a key part of the ribosome, helping to read the mRNA and guide the assembly of amino acids into a protein chain. And how does this work? Translation starts when the ribosome attaches to the mRNA. The ribosome reads the mRNA in sets of three nucleotides called codons. Each codon corresponds to a specific amino acid. Anticodons on the tRNA match up with the codons on the mRNA, ensuring that the right amino acids are brought to the ribosome. The tRNA then brings these amino acids, which are linked together by peptide bonds, forming a growing protein chain. Think of translation like assembling a necklace. The mRNA is an instruction sheet with a pattern of beads, codons, and the tRNA acts like a delivery truck, bringing the correct colored beads, amino acids, to the assembly line, ribosome. The RNA is like the workers in the factory, making sure the beads are strung together in the right order. As each bead is added, the necklace or protein grows longer. The process stops when the last bead is added and the necklace is complete. The peptide bonds are the links that hold the beads together, forming the finished product. Normally, when the TRPV4 protein is made correctly, it helps form strong cartilage that holds the ears upright. But what happens when there's a mutation, a permanent change in the DNA that happens due to errors in DNA replication, environmental factors like radiation or chemicals, or inherited genetic changes? That's where the mutation in Scottish folds come into play. Let's look at the mutation in more detail. The normal TRPV4 protein has this DNA sequence, which is translated into the amino acids, but in Scottish folds, the mutation changes the DNA too, leading to the amino acids. This small swap from cytosine to thymine is a point mutation in the TRPV4 gene, specifically a missense mutation. This means that one DNA base was changed, leading to a different amino acid in the protein sequence, causing a weak cartilage. This mutation affects more than just ears. It can also lead to a painful condition affecting the joints and bones. In some cats with two copies of the mutated genes, the effects are severe, causing joint deformities and difficulty moving. Even those with one mutated copy may develop arthritis as they age. Though Oliver's folded ears are undeniably charming, the mutation comes with serious health concerns. This condition weakens cartilage not just in the ears, but throughout the cat's body, leading to joint deformities, arthritis, and difficulty moving as they age. As the breed becomes more popular, the risks of these painful conditions have sparked important conversations about breeding practices. Understanding the science behind the Scottish Fold mutation not only deepens our appreciation for these adorable cats, but also highlights the importance of ethical breeding and the responsible handling of genetic traits. The next time you meet a Scottish Fold, remember it's more than just a fold of the year, it's a story written in life's genetic code.